So now we're looking at this is 2018. I think this is section A, right? So the module one for section A. And what we're generally looking at now is describing the chemical structure, right, of the phospholipid, right? So the chemical structure here, we're gonna have our phosphate group, right? So let me draw the chemical structure. Some persons are saying that we could just use a circle and a rectangle and just draw the tails, right? But some persons are being bothered by the fact that it says chemical structure. But in that case, it shouldn't be too hard to produce our chemical structure, right? We know that we have a phosphate there. We set on all sides by oxygen. We're gonna have a side negative oxygen here. We're gonna have a ester group combined to the R linkage here for the choline group, and then we're gonna have an oxygen there. It's gonna be binding here now to a CH2, right? One to a CH. You guys know the structure of these things from biochemistry, so it shouldn't be too hard to produce a glycerol molecule. Shouldn't be too hard at all. Then we're gonna have an ester group, ester linkage now. And we know how ester linkages look from CSEC chemistry. Remember, this is just biochemistry, right? Biochemistry is nothing but the application of chemistry in biological systems. And then you'll have your fatty acid chains, right? So yeah, it doesn't look too good. That's because I'm drawing it here. But you know, you just draw it small enough for it to be encompassed within the box. And here we have the structural components, right, of the phospholipid. So you have your phosphate group, and the R group would basically be the choline structure. You'd have your glycerol as well as your fatty acid tail or chains. All right, that should be lovely. Describe how phospholipids oriented in bilayer. So how are they oriented within the bilayer? Go ahead.
Sir, do we actually have to draw out the chemical structure? Could we just write the names, the circle, and the tail thingy? And
Okay, so yeah, all right. So when I was doing this, some person when I did that first, right, initially, some persons noted out that it says chemical structure, and they were feeling that okay, we should provide a chemical structure, right? But it shouldn't be that hard to remember the chemical structure. So I think if they ask a chemical structure, it would be best to provide this, right? And then you could use a ball and sticks to show what the chemical structure would be looked at on a simplified way. So it says chemicals are on. I took the concepts into consideration and actually put the chemical structure. If you get what I'm saying, because the word chemical is there, it bothered some people and wondered whether or not they should just draw the circle and stuff like that. But the chemical structure is there, shouldn't be a problem. Because you guys would have looked at lipids as well, so you should be able to draw a, a glyceride without any type of issue with ease. So all you have to do is just draw the phosphate group on the R on top, right? So we have that there. Oh, we're supposed to talk about what is hydrophilic and what is hydrophobic. Which part is hydrophilic and which part is hydrophobic? The head is hydrophilic, right? So you have a hydrophilic head. The head is hydrophilic, and then we have a hydrophobic tail, right? Lovely. So how are they gonna be oriented now, to form lipid bilayers? How? Do you guys want me to just go through the questions then? Because I'm not sure if it will take a lot of time to wait for persons to answer. All right, so let me just go through them, right? So you have a lot of space on your paper, right? A lot of persons have been complaining about how are they gonna be explaining these things. There is something here called space, and within this space you can draw a little diagram to show what you're talking about. You can always draw your little lipid bilayer and then talk about the diagram and talk about the bilayer, right? So we know that they would have the hydrophilic heads facing out towards the extracellular environment and inwards towards the intracellular environment, right? And then within the membrane itself, it is the fatty acids turning away from the aquatic environment, right? Lovely. Extracellular, intracellular. use the terms. In biology, specificity is key. Alright? Specificity is your friend, but it must be the correct variation, right? So Justin sent something in the chat. So inside of the cell, right, the cytoplasm contains lots of water, and so does the liquid outside. So the hydrophilic heads of the phosphate molecules is attracted to the water due to tiny charges, right? It contains, right, because you know the phosphate group, right? It contains oxygen, and those are polar components, so it will be attracted to the water, right? While the hydrophobic tails would be repelled by water, causing the phospholipids to arrange themselves in a double layer, with heads facing outwards towards the extracellular and intracellular environment, and the tails facing inwards to create that phospholipid bilayer. Lovely. So that is a lovely response in the chat there by Justin. Right? I'm not sure if I could use that response. Alright. Hoping it's no issue, Justin. I'm just going to use that response here and place it on the screen. Alright. So that response is a lovely response to the question. It is the best response to this question here. Alright. That I've seen in a while. Alright. So that is the best response that we have there to that question. And you can, if you lose your way, you could always use the space on the paper and draw your diagrams. They can't stop you, right? And they will see the diagrams when they're marking, so they'll know that you know what you're talking about, even if you mess up somewhere within sentence, right? Try to help yourself to get the best marks. All right. State the name of the lipid. Is it lipid responsible for maintaining fluidity of cellular membrane? What would that lipid be? Quickly name that lipid. Go ahead, Jelena.
So the cholesterol.
Cholesterol. Right? Why? Oh, I put the R in the wrong place. You see? Cholesterol, right? Lovely. Describe how intrinsic proteins allow for the movement of substances across cellular membranes. Now you can use this diagram as well to show a little bit of the phospholipid membrane and bilayer specifically, right? And then you could show your membrane, transmembrane protein. All right. So how? Somebody describe it, right? How is it done? So I would start by saying that our membrane itself actually prohibits the movements of certain molecules, right? So it prohibits the movement, right, of certain molecules such as large charged anionic structures as well as large non-polar structures. They are not going to be passing through the membrane. So our transmembrane, our integral proteins, right, our intrinsic proteins, right, would actually be used as pathways through the cellular membrane. So you have are channel proteins and you have carrier proteins. Channel proteins generally help with facilitation of facilitated diffusion, and our example would be the aquaporin. Facilitated diffusion is with is along the concentration gradient, and then we have things like the channel proteins, right? The most notable would be sodium potassium pump, right, which can actually use energy, so it's active transport, use energy to actually move our charge and large molecules across the membrane that otherwise could not pass through the membrane. So you could speak about how those things will allow the movement of substances across cellular membranes using these intrinsic proteins. Right? So I would love for you guys to speak about those stuff. It should be fine. 4 marks. That should be lovely. Alright. Let's look at this structure now. Identify the class of lipid here. This is not a steroid. It doesn't look like a steroid to me. So I think this would be a triglyceride. Right? And we know that since it's a triglyceride, now we're looking at the 2 structural components that would actually speak to its function within the animal tissues. So we know that it's an energy source. Right? And that's because it has a high level of C bonds, C bonds, right? High amounts, should be high amounts. of C bonds, right? That would speak to the energy source component, right? The thermal regulation component would have been to its large molecular structure, right? It is very insoluble, and what's characteristic? It is a triester, and esters are insoluble. Large chain esters are insoluble, right? Speak to those type of things. It's biochemistry, and I'm not hearing the chemistry from the biochemistry. So speak to the chemical structure, right? So they're insoluble because they're large esters. Esters are generally insoluble, right? And that assists with the temperature regulation of the use of the human body, right? And because it's large, right, and creates these globular structures, right, it can be used as shock protection, right? So it can be used as a physical protection for internal organs and specifically the skeletal system as well. Mm-hmm. Mm-hmm. It has other functions as well, but we can't specifically refer to the structure in order to describe those other functions. But you guys should be able to get the function of a triglyceride there. That should be fine. 2 marks too. That should be lovely. Right, and that should head us into the module 2 component of section